Welcome back to the deep dive. Today, we are strapping in for a really fundamental topic. We're looking at the absolute foundation of your existence,
We really are.
the ultimate biological energy transaction that fuels every single process in your body. We are talking about cellular respiration.
And we are not just looking at the biology. We're going to examine the fundamental physics and, the chemistry of how life masters this kind of controlled combustion, how we harvest potential energy from, well, the most common biological fuel, glucose.
And our source stack for this is pretty wide, isn't it?
It is, yeah. It ranges from these high-level academic outlines that connect these cycles to, say, ecosystem dynamics, right down to some really advanced papers detailing how engineers model these microscopic energy flows using concepts like bond graphs.
So our mission is to give you the complete picture. We're going to slow down and unpack the three essential stages of aerobic respiration. We'll look at the ingenious workarounds life uses when oxygen isn't available, and then reveal some of the surprisingly elegant techniques scientists use to actually measure this stuff.
To really give you a serious appreciation for your body's efficiency. Exactly. So, to start, where does this all fit in the grand scheme of things?
I think the best way to frame it is as the reciprocal partner to photosynthesis. Photosynthesis is the builder. It captures light energy, uses CO2 and water to create glucose and oxygen.
And cellular respiration is the dismantler?
Precisely. It takes that glucose and oxygen, breaks them back down into CO2 and water, and in doing so, it captures a colossal amount of stored energy.
It's a massive exchange. I mean, when you look at the raw chemistry, the complete oxidation of 1 mole of glucose releases 686 kilocalories of energy.
And that complementary cycle is why the entire global ecosystem, from the bacteria in the soil to you listening now, is sustainable. It's this continuous, self-sustaining loop.
What the plants put out, we take apart. So let's bring it down to the personal level. I think one of the first things we should probably clarify is the common confusion between breathing and cellular respiration. They're not the same thing. No, not at all, though they are linked, of course. Breathing is the physical act, the gas exchange. You inhale oxygen, you exhale carbon dioxide. That all happens in your lungs.
Right.
Cellular respiration, however, is the actual aerobic harvesting of energy that happens at the cellular level. This is the multi-step chemical reaction, mostly inside the mitochondria, the power plants of your cells.
So the oxygen you breathe in is really just the critical ingredient for that chemical engine to run.
That's it, exactly. And the goal of that whole machine is to translate the energy stored in glucose into the cell's universal currency, which is ATP.
And this is what scientists call an exergonic process, meaning it releases energy.
Yes. And the cell is remarkably good at capturing it. When transferring that energy from the glucose bonds to ATP, the cell manages to capture approximately 34% of the glucose's original potential energy. The maximum theoretical yield is about 32 ATP molecules per glucose.
32 ATP molecules. I know that number can vary a little, but that level of efficiency is, well, it's just mind-blowing.
It is. And that tiny microscopic process is what pays your entire energy bill. The average adult needs something like 2,200 kilocalories a day.
And it's important to remember that calorie you see on a food label with a capital C, that's actually a kilocalorie.
Right, a kcal. It's the heat needed to raise the temperature of 1 kilogram of water by 1 degree Celsius.
That really makes the numbers feel much more tangible. We're literally measuring the potential heat energy in our food. And where does the bulk of that 2,200 kilocal go? Is it all for moving around?
You'd think so, but no. Actually, 75% of your daily intake is just for essential maintenance. Keeping your heart beating, lungs moving, nerves firing, basic life support.
Wow.
Only about 25% is left over to power-conscious physical activities.
And that 25% can vary wildly. The sources give some great examples.
They did. Take a 67.5 kilogram person. Sitting and writing might burn, say, 28 kilocal per hour. But running at a fast pace, 8 to 9 miles per hour, that demands 979 kilocalories per hour.
So the cellular engines go from idle to full throttle almost instantly. So let's look under the hood at those engines, the three main stages. If we strip away the jargon, the basic idea is physics, right? It's about electrons falling.
That's the perfect analogy. Electrons are pulled toward oxygen, which is highly electronegative. It has a really strong pull. Think of it like a controlled waterfall.
If you just let the water drop all at once, you get an explosion.
Exactly. Cellular respiration lets the electrons fall step by step, releasing energy gradually so it can actually be captured. And this movement of electrons defines the key reactions.
The redox reactions, oxidation and reduction.
Right. In the overall process, Glucose is oxidized. It loses electrons and hydrogen, eventually becoming CO2. And oxygen is reduced. It gains those electrons and hydrogens, forming water.
Okay, so stage 1, glycolysis, the universal first step.
Right. Glycolysis happens in the cytoplasm, so outside the mitochondria. We start with one 6-carbon glucose molecule, and we split it into 2 3-carbon molecules of pyruvate.
And it has 2 phases. First is the energy investment phase. Why does the cell have to invest energy using 2 ATP just to start breaking down glucose? That seems a bit backwards.
It does, but it's a bit of biochemical sleight of hand. The cell uses that initial investment of 2 ATP to make the glucose molecule unstable and trap it inside the cell. Once it's unstable, that 6-carbon ring easily splits.
Ah.
Then comes the payoff phase, which yields a total of 4 ATP, so a net gain of 2. And crucially, it also reduces 2 NAD+ molecules into the high-energy carriers, NADH.
So we've made a tiny bit of ATP and these important electron carriers. Those 2 pyruvate molecules then have to get into the mitochondria for stage 2.
That's right, in eukaryotes at least.
Yeah.
The pyruvates first undergo a grooming process, pyruvate oxidation.
Yeah.
Each one loses a CO2 molecule, and the remaining 2-carbon fragment joins something called coenzyme A, creating acetyl-CoA.
And acetyl-CoA is the molecule that actually feeds into the citric acid cycle.
The Krebs cycle, yeah. This is the factory that finishes dismantling the original sugar. Acetyl-CoA combines with the 4-carbon compound to form citrate. And through a series of steps, the cycle systematically breaks citrate back down to that starting compound, releasing 2 CO2 molecules along the way.
And since we started with 1 glucose, that means the cycle turns twice.
Twice. And the goal here is really carrier production, not bulk ATP.
Right.
Over 2 turns, we get 2 ATP, but we also get 6 NADH and 2 FADH2. So if you're keeping score, we've only made 4 ATP total so far. The vast majority of the energy is now locked up in those electron carriers.
Which means everything has been a preparation for stage 3. Oxidative phosphorylation, this is the grand finale.
This is the powerhouse. It involves 2 parts, the electron transport chain, or ETC, and chemiosmosis. NADH and FADH2 deliver their high-energy electrons to the ETC, which is a series of protein complexes embedded in the inner mitochondrial membrane.
This is where the electrons finally start their cascade down toward oxygen.
That's right. As the electrons move down the chain, they drop energy in a controlled fashion. And oxygen is waiting at the very end to accept them, combining with hydrogen ions to form water. But the energy that's released isn't used directly to make ATP.
No, it's used for something else.
It's used to pump hydrogen ions, protons, really, from the mitochondrial matrix into the little inner membrane space.
And pumping all those positive ions into a tight space creates a huge amount of stored energy.
It creates what's called the proton motive force, or PMF. It's highly energetic because it's actually 2 gradients at once. A concentration gradient, right, more protons on one side, and an electrical gradient, a charge difference.
That PMF sounds like a battery that's just ready to discharge. How does the cell harness it?
It lets the protons rush back across the membrane, but only through one specific channel, this incredible protein machine called ATP synthase.
And this thing acts like a tiny generator.
It literally does. The flow of protons through it rotates parts of the enzyme, and that physical rotation drives the synthesis of the vast majority of the ATP, yielding up to about 28 molecules.
The whole system is so elegant, but also incredibly complex, which must make it vulnerable.
It is. Very. Any interference with the ETC is catastrophic. Poisons like cyanide and carbon monoxide, they bind to the electron carriers and just halt the electron flow instantly.
And if the electrons stop, the pumping stops, the PMF collapses.
And the cell starves of ATP. It's that fast.
But there's a stunning evolutionary cheat code where the body intentionally reduces that efficiency, the whole brown fat mechanism.
That's one of the coolest details. In brown fat, which is really important for newborns and hibernating animals, there's an uncoupling protein. It essentially punches a controlled hole in the membrane, letting the protons leak back without going through ATP synthase.
So it bypasses the generator completely.
Exactly. And instead of capturing that potential energy as ATP, the cell releases it instantly as heat. It's intentional energy inefficiency, but it's vital for non-shivering heat generation.
That is just amazing. So let's consider the alternative. What happens when that crucial ingredient, oxygen, just isn't there, say during a sprint?
That's when you shift to fermentation, the anaerobic pathway. It relies only on that ancient first-stage glycolysis, and it yields a tiny 2 ATP per glucose.
It's a massive drop in efficiency. If it's so low, why even bother? What's fermentation's real purpose?
Its main goal isn't really ATP production, it's regeneration. Glycolysis needs a continuous supply of NAD+ to accept electrons. When oxygen is gone, the EDC stops, and all the available NAD+ quickly gets stuck as NADH.
So the system gets clogged up.
It gets clogged up. Fermentation is just a trick to oxidize NADH back to NAD+ so that glycolysis can keep spinning, securing that tiny, life-sustaining trickle of 2 ATP.
And this leads to two familiar byproducts we all know.
Right. Lactic acid fermentation happens in your muscle cells when you push them hard, and oxygen can't keep up. Pyruvate is reduced to lactate. Same process gives us the tang in yogurt and sauerkraut.
And the body is clever. That lactate doesn't just sit there.
No, it's shipped off to the liver, where it can be converted back into pyruvate once oxygen returns.
And the other kind is alcoholic fermentation.
Used by yeast, yeah. Yeast converts pyruvate into CO2 and ethanol. That's what makes bread rise and what gives us wine and beer. But both forms are just serving the same basic function: keep glycolysis running.
It's powerful how universal glycolysis is. It seems to be in every living thing.
That universality is one of the biggest takeaways. Since glycolysis happens in the cytoplasm and doesn't need any complex organelles, it's strong evidence that it evolved very, very early, maybe 3.5 billion years ago, long before the atmosphere had free oxygen.
So it's a piece of evidence for the common ancestry of all life.
A really strong piece.
We've focused on glucose, but the body is highly adaptable. We don't just run on sugar.
Absolutely not. Cells are incredibly flexible. They can feed carbohydrates, proteins, or fats into the cycle. But when we look at pure efficiency, fats are just superior fuels. Gram for gram, they yield more than twice as much ATP as carbs or proteins.
Why is that? Why are fats such a better fuel source?
It comes back to the physics of electrons again. Fats are essentially long chains of hydrocarbons. They are packed with energy-rich hydrogen atoms and their electrons. These molecules are almost entirely reduced, so they have a huge store of electrons ready to be stripped off and fed into the ETC.
A much denser, more efficient energy package.
Exactly.
That really puts the energy density of a fat molecule into perspective. Okay, let's pivot to the practical science. If you're a researcher, how do you actually measure the rate of cellular respiration for something small, like a sprouting seed?
The standard method is using a device called a respirometer to measure oxygen consumption. You can also measure CO2 production or heat release, but O2 consumption is usually the most straightforward.
And this involves that famous potassium hydroxide, the KOH trick, to make it work.
It's absolutely necessary, because in the overall reaction, for every molecule of O2 you consume, you produce 1 molecule of CO2. So if you just sealed an organism in a container, the gas volume wouldn't change at all. You couldn't measure anything.
So the goal is to get rid of the CO2 as it's produced.
Precisely. You add potassium hydroxide, KOH, to the chamber. The KOH reacts with the CO2 and turns it into a solid salt. By removing that gas byproduct, the consumption of oxygen now causes a measurable drop in the total gas volume.
And that drop in volume directly tells you the rate of respiration. But environmental factors are always lurking to mess up your experiment. I know respirometers are notoriously sensitive.
Oh, highly sensitive. And that's where the physics comes in. Respirometers are governed by the general gas law. Pressure, volume, and temperature are all linked. If the room temperature goes up by just 1 degree, the gas inside will expand, making it look like the organism is consuming less oxygen.
When it's really just the physics of the environment changing, so you need a control.
You absolutely need a control to separate the biological change from the environmental noise. The control respirometer is identical, but instead of a living thing, you put in something that doesn't respire, like glass beads or boiled seeds.
So any change in the control is purely due to temperature or pressure shifts?
Exactly. You then use that control reading to correct your experimental reading, and that isolates only the volume change caused by the organism's actual oxygen consumption.
Okay, let's end by touching on the advanced modeling. When we talk about protons and electrons moving across a membrane, it sounds less like biology and more like electrical engineering.
And in many ways, it is. Scientists and engineers use these sophisticated energy-based models to tackle the complexity. A powerful example from the sources is the use of bond graphs, which treat the biological system like a circuit or a mechanical system.
How does that work? How do you apply circuit physics to a mitochondrial membrane?
Well, bond graphs use universal physical concepts like flow, effort, resistance, capacitance, and they apply them at the molecular scale. For instance, the proton motive force, that huge gradient, is modeled as a form of potential energy storage, like a capacitor in an electrical circuit.
And friction or heat loss is just resistance.
It's modeled as resistance, yes. You're creating a simplified but physically plausible machine model of the ETC.
And this kind of modeling actually confirms the biology.
It does. For example, by modeling the complex chemical steps, these simulations physically demonstrate that the net effect is the pumping of about 20 protons across the membrane for every NADH molecule used. It confirms the physics driving the PMF and the overall efficiency we were talking about earlier.
That is the complete deep dive into the cellular engine. We've gone from the global carbon cycle all the way down to the submolecular physics of the ATP synthase generator. We've seen that life's primary energy strategy is built on this incredible controlled efficiency, harvesting about 34% of the potential energy in glucose.
And we learned that this core process relies on glycolysis, the ancient, universal energy mechanism that works without any organelles at all.
Which leaves us with a provocative final thought. If the electron transport chain is capable of generating up to 28 ATP and relies on all that complex, precise machinery, what fundamental evolutionary trade-offs compelled early life and continues to compel all life to maintain glycolysis, that low-yield 2-ATP pathway, even after the massive payoff of oxygen-based respiration became possible?
Why is that simple, inefficient, ancient pathway still the universally conserved bedrock of all biological energy generation? It's a powerful question about survival, redundancy, and the deep ancestry we share with every living thing.
Something to mull over until our next deep dive.